there's a lot of information online. A lot more has appeared even over the last 5 years, 6 years since I've been in this work. You know, NRCS is coming out with a lot more about the soil food web. If you go to my blog, www.thermalcomposting.wordpress, you'll find a resources list of lots of books that you can read. YouTube videos are helpful to start hearing the conversation and the vocabulary that's being used. But I think something that's really important is that people need to go outside and they need to bring a shovel and they need to look at their soil. They need to dig down in it, they need to smell it, they need to touch it, they need to put their hands in it. What do the roots look like? How deep are they going? What kind of organisms are they finding? And if you have access to a microscope, a local high school, local community college, start looking at stuff. Many of the products that people send to me that they want for me to endorse or for my mentor, Dr. Lane Ingham, to endorse, there's a few different kinds of bacteria. Oh no, 5,000 different species of bacteria. Cool. You can make that in your vermicompost easy. So start looking at these things that people want for you to buy or that you're creating. And if you don't know what to call it, that's okay. But just start becoming familiar with what it looks like. Because then when you start applying biology to your land, whether it be in the form of compost or teas or extracts or animal manure or four-legged critters that are grazing in a different way, you can see the changes that are happening at that microscopic level. And that lets you know that you're moving in the right direction or that you're not really moving in a direction at all. Does anybody else have some suggestions for resources?
I would suggest that it's important for me to hire somebody like you, or Elaine, or Nicole Mass, somebody, because I think it's really good to do things exactly what they're talking about, but try to do things on a scale. I don't want to, unless you really are science-minded, or have room for all that information, is probably, I think you need more knowledge, professional knowledge, to try and do things on a bigger scale.
What's your name?
Roger.
Thanks, Roger. I think that's a great suggestion. I mean, it works for me. But the other thing is that I do have an interest in putting myself out of a job. You know, by the end of this decade, this knowledge should be common knowledge. If you can identify the difference between an apple, an orange, and a banana, you can begin to identify the difference between bacteria, fungi, protozoa. I'm not saying that all of you need to have this skill. Not, you know, thank goodness for diversity of people and jobs. But I'll tell you what, the people that eat this up are little kids. And their eyes are really good. And they're happy to make a few bucks, right? If you can identify the difference between bacteria, fungi, protozoa, and then you can count, well, then you finish your spreadsheet. And now you have your fungal bacterial ratio, and you understand what kind of nutrient cycling you have going on. But absolutely it helps. And one of the things that I encourage communities to do, if it doesn't work for one individual's resources to have me come out and work one-on-one with them, then get a group of 5 or 10 or 15 people together. Let's spend 2 to 3 days together. A lot can happen in that time. And then we keep in touch. A lot of farms that I go to, I go just once a year and make sure that things are on track.
Molly, if you're a start, just wanting to do something, start building on compost, what would be your first suggestion?
There's so many different ways to make compost. If you want to make compost quickly, and you want to make sure you're killing weed season pathogens, then you need to learn how to do aerobic thermal composting. If you don't have the physical capacity to turn a compost with your hands and you don't have access to the machinery, start exploring vermicomposting. But I think that if there's one thing that you come away with today, in terms of knowledge, remember that diversity creates resiliency. And we have major fluctuations in temperature and the hydrological cycle now. And so when you have a diverse community of organisms, and I'm not just talking about microscopic organisms, I'm talking about plants as well, or ingredients in your compost, that's going to help you out. That's a very basic way of answering that question. But there's a lot of videos online, again, YouTube videos, compost workshops. But if you go back to the conversation that we had earlier about plant succession and the type of plant foods that organisms consume, like bacteria like simple sugars, and fungi like more complex carbohydrates, then if you want to make a product that's more fungal dominated, add in more fungal foods. If all you're adding into your vermicompost is kitchen waste, well, it's quite likely you have a bacterial dominated vermicompost. And you probably have other organisms too, but add more carbon material, shredded paper, fall leaves, straw.
I was wondering if you did want to read a little deeper. I was trying to do the Google thing, but you come across articles that they like $30 to get them. Is there a good journal out there to follow or not?
Oh, that's a really good question. Off the top of my head right now, there's not one that I've been going to consistently. It's kind of been all over the place. But there are a few on the resources page on my blog. What specifically are you Googling?
All of the above. I could benefit from more of a focus, but I've just been looking at everything from compost to different cover crops to different grazing methods, winter grazing ideas, lots of stuff. So yeah, I was just wondering if there was a soil health agriculture related journal that was like that condensed a lot of it well, or if it was just kind of piecemeal.
In my experience, it's been quite piecemealed, and one of the things that's happening in soil research, it's beginning to change, but you know, to have something published in a scientific journal and to put together a research study, you have to isolate variables. And who in this room has the ability to isolate variables in their growing system? Right? So if I'm creating, or I'm replicating a certain species of bacteria that can offer my plant more nitrogen or phosphorus or magnesium or potassium or whatever, and I'm growing this out in a laboratory, and my laboratory is air conditioned, and you know, I have this particular environment in my laboratory and I can grow these bacteria there. Let's say I'm in Southern California or something. And now I take this organism set, oh, and I've done my test plots, right? So I've done my test plots and we're outside of Southern California somewhere. So I have these results and it's really great. And now everybody needs this because now you can get phosphorus into your plant. And then I take this bacteria and I bring it up here. Do you think that beauty queen is going to be able to handle it? So this is where regenerative agriculture is really important, because if you've seen one farm, you've seen one farm. Is it on the north slope or is it on the south slope? Did the ingredients come in with chemicide residue or did they come in organic? Do you have 60 years of chemicide residue in your field, or has it been organic forever? It's different everywhere. So this is where citizen scientists are really important, because only recently has this work began to appear in major universities. And it's really challenging to isolate the variables, and why, if we want to apply this in the real world, do we even want to isolate variables? In the world of soil microbiology, 1 + 1 doesn't equal 2. Sometimes it's 3, sometimes it's 7, sometimes it's 0. So my interest is that the basic principles of looking at who eats what in terms of fungi and bacteria, and that you can get the fertilizer bag openers into your soil, and however you can get them in, and it helps to know what's in your soil to begin with, so that whatever it is that you're applying, you know that it's making a change. And if it's not making a change, then we have to start doing some other kinds of analyses to find out. But it's pretty rare that changes don't happen, even with the addition of just a bacterially diverse bug in a jug kind of a thing. Does anybody else have something to add to that question? Will you pass the mic?
I don't know if I'm adding a question or not, but I'm just wondering if you had an opinion more specifically about changing the soil microbiology from cover crops. Did you attain that rather than compost and spreading, building the compost and spreading the compost? Did you attain these objectives through planting various species of cover?
That's a great question. Did everybody hear that?
Maybe you could repeat the question.
Okay, the question was, can you create a diverse soil microbiology using cover crops instead of bringing in compost and compost tea? Yes, you can. How much time does it take? Like we said at lunch, standard ecological answer, it depends. Where are you? What's been happening to the land? And how much time do you have? If we have 60 years of ag left in our soils worldwide, well, maybe not that much. But you always have to go back to your time, or your resources of time and equipment and finance, right? So if cover cropping is the way for you to bring in a diverse microbiological set, that's important. There's not one tool, right? We need a big toolbox, and we need access to lots of different tools, because biology doesn't do anything on its own. Like Blaine was talking earlier, you need the soil covered. Why? Well, you need plants there, and you need organic matter to feed organisms. Organisms need food, just like you and me. So that cover crop is really important. And you will start to build a biological set. Do you have 20 years to do it? Maybe. That's great. It really depends on what you have the ability to do, but absolutely cover cropping, especially diverse cover cropping. Let's think about that beautiful image that never gets old to me about pasture plants and their above ground parts, and you can see their below ground roots, and how Blaine was talking about how each plant contained a different mineral set in it. So when you develop your cover cropping systems, you're looking at diversity of species, what's going to grow in a drought year or a deluge year or cold season or warm season, and look at different root depths and organic matter biomass and functions, right, like nitrogen fixing plants.
I've been doing no-till for about 20 years. We got about a couple thousand acres in it. And we've gone from about less than 1% of organic matter in our soil. That's when we pinned up after a lot of conventional farming. And now we're up to 3%. And the earthworms are coming back. And we kind of tell when they come back, and because the yields will go up like
Are you doing any sort of surface cultivation or shallow? Okay, so pure, for real no-till. And how long is it taking for your organic matter layer to break down?
It goes very fast. There's not as much as I'd like to have next year. I mean, it seems like it's gone too fast, but it's gone.
Okay, I would consider looking into a permanent understory or a permanent cover crop and bring in fungi. The nitrogen that you're adding, what form of nitrogen is it?
It's not ammonium nitrate, it's the urea.
So our weed seeds respond to nitrates, and essentially you're feeding bacteria, some species of bacteria that can handle this application, and then you're also prompting weed seed germination through this form. But bring in fungal inoculation in some way that you can, and that might be inoculating your seeds when you're planting your seeds, so that as the roots grow down, the fungi can grow down along with it.
So how do you get, would this be kind of like a compost tea that you put on the seed?
Yeah, there's a number of different ways you can go about it. There are products that you can purchase. I don't know of any off the top of my head that I could give you a name for, mostly because I encourage people to source something locally so that we know that it can survive in that environment. But you can inoculate seeds in the form of rolling them in a compost or spraying them with a compost extract or a compost tea.
Yeah, we've done that a little bit before. There's this compost that releases phosphorus in the soil or bacteria if you lie, and you basically turn it into a tea and then you put it on seed as you're planting it.
Right, so these products, when you make them, it's important to look at the product and who is there. A lot of the work, the research that's been done on microbiologicals, the tests are done in the form of a plate count. And so what that means is you grow out a set of organisms that are eating a certain kind of food, let's say phosphorus. And so you grow them out on a plate, you put it in, you give them a certain type of food, and you put them into a certain type of environment to grow. Is this the result of what you're actually brewing? Well, that could be a part of it, but the tea brewing process is so dependent on your brewing equipment, the water that you have, the temperature that's there, what kind of products you're putting in, that in my experience, the only way to know what you're actually spraying out is to look at it and see if it's there. And that's one of the most beautiful things about this method of looking at soil and soil products. It's a qualitative assessment, not quantitative. It tells you the quality of the product and are all of your apples, oranges, and bananas there. And if there's just a little bit there, but at least it's diverse, okay, well, it's probably better than what's in many fields. So that's a good start. Does anybody else have anything to add to this gentleman's question?
I've heard that, you can tell me if this is true, that fish hydrolysate might help with cheatgrass.
Fish hydrolysate is a fungal food. So I don't know that it's directly the fish that is keeping the cheatgrass at bay more than it could be the fungi, the fish that's feeding fungi in the soil that inhibits that particular plant species to grow. And there are certain types of bacteria that assist in the growth of cheatgrass. I haven't put it up on my blog yet, but based on that question, I'll get it up by midweek for you all, so you can check that out. But again, it's an isolated species of bacteria. One of the things that's so cool about making a compost with a capital C versus one with a lowercase c, so we're talking about quality versus quantity here. There's particular approaches to making a high quality compost product, And I'd be happy to talk more with you about that. Also, if you find Richard, pick his brain about it as well. He's learned the ropes. And so we behave differently in isolation than we do in consortium. Okay, so when you purchase a product that's just one particular type of bacteria, like this kind that can inhibit cheatgrass, how does it affect the other microbial community? And also, are you setting some things out of balance? Here's an example story. Trichoderma. Has anybody heard of this fungi before? In the laboratory, trichoderma was found to consume non-beneficial bacteria. In this particular study that was being done that prompted trichoderma to go out onto the market, they never tested what trichoderma did to beneficial bacteria, beneficial fungi. So let me ask you something. What's your least favorite food?
Okra.
You're in good country if you don't like okra.
I could not say that in the South.
Right, yeah. Well, you're far from it today. Okay, so let's say you're in jail and the only food that's being brought to you is okra. Would you eventually eat it?
Yes.
Yeah. And remember that beneficial and non-beneficial organisms, for the most part, are just like you and me. They need food, air, water, shelter. So if you're hungry enough, you'll eat something. And so this trichoderma, put in these isolated plates, fed, or in an environment with only non-beneficial fungi, that's what they ate. But now we know that when we take them out of isolation, we put them in other soil systems, that they don't only eat non-beneficial fungi, they also consume beneficials. And so we're messing with the balance, and we have to be responsible about this. I'm not saying don't buy it. I'm not saying it can't be something that can be utilized. I'm saying think about what we do to our land and what we bring into our land in terms of tree years or geological years instead of 60 years. So there's a lot on the market, but what you can develop in your composts in your area is going to be diverse, and also the organisms create their own community. And so in a sense it stays in balance that way, and it's more safe to apply into the land. Yes, sir.
I'm just thinking of a methodology. If you've got a piece of ground, let's just use cheatgrass for example, and you've got a piece of ground and half of it's got cheatgrass and half of it doesn't. Can you, through these methods of measurement of the microorganisms in the soil, go to where you don't have cheatgrass, and you do a measurement of what's there, and assuming you got similar environment and temperature and whatnot, and go over and say, okay, this is the end product I need over here where I got cheatgrass.
So what you're saying is we're looking at productive versus least productive. Can I see
I'm just
that?
using cheatgrass as a species you're trying to get rid of, and you've got the same piece of ground, and some of it has cheatgrass and some of it doesn't. Is that a valid methodology, rather than go over here to the cheatgrass and say, okay, I'm going to test this and figure out what's there and what needs to be added? Can you go to a place where it's not and figure out what needs to be the end result?
What microbial community is there? Absolutely, you can look at that. But biology is one part of the story. That's talking about our living plants and our soil microbiology. There's also the parent material of your soil, your sand, silt, and clay, and then there's the chemistry relationship in all of that. So my recommendation is let's target biology, and as productivity increases, if we're starting to see a change isn't taking place, let's consider assistance in change of chemistry. And the reason why I often use that as a last resort is because if you mess it up, it can be very expensive and very hard to fix. But certainly when I go out to a growing system, we'll look at most productive, least productive, and you could even send me samples and not tell me where they're coming from. That's what I prefer. And then we look at the data, and then I tell you what I see, and then you can say, oh, well, yeah, that's the area with the cheatgrass and that's the area with my productive pasture.
Well, why I was asking about that method is that it sounds like there's a whole bunch of variables. I mean, that's an understatement. So if you take an environment that's close to the environment you're trying to correct and define those variables the way they exist in whatever you define as healthy, it would seem like it would cut down the process time or the experimentation you'd have to do over in this other area.
Are you talking about bringing in the biology from that other productive area and bringing it into your non-productive area, or your cheatgrass area?
Yeah, I mean, like you were talking about fungi in particular. If you went over to this other area and the counts were high and the method was appropriate, would that save you some time and expedite things?
Oh, sure. You could absolutely take some of that soil and brew it out and apply it onto this other area. Maybe you could even consider, if you were going to replant that area with cheatgrass, or that has the cheatgrass, to use the native soil microbiology, inoculate your seeds with that in some way, and then plant them. One more question.
I just want to add one comment. I believe it's critical to manage for a healthy whole. When we try to manage against the species, we will always fail. You have to manage for a healthy whole, and the problems will disappear to a large extent. Think about the principle. You got to capture solar energy, you have to have diversity, you have to have an effective water cycle, and all those bugs and mollycops and what we're supposed to do. I think that's key. You know, it's human nature, people. That's a bad one. You got to get rid of it. Worry about making everything else better, and that bad one will disappear.
Logan?
As far as personal individual next steps, if you're going to undertake any of the steps we've talked about, do you have a short list, or do you have a short list of, if you're going to monitor changes, what would be your most important things to monitor before you ever start? Yes, what would be the most important things to monitor before you get going?
That's a great question. Does anybody want to pitch in on that before I go into it? Because I'd love to hear from y'all.
I was just going to ask you to use the menu since you're talking about monitoring.
I don't. But that doesn't mean you can't. But I don't. CO2 respiration from who? Fertilizer bags? Or fertilizer bags and fertilizer bag openers and spreaders?
Well, I guess if you already knew that having an application on commercial fertilizers, and you were just simply sampling whatever microenvironment in existence.
Yeah.
Right?
Just look at the critters.
You don't know what you got to look at.
So things that I think are important, obviously productivity. So if it's cattle, that's your harvest, if you will. What's their health? What's their weight gain? What's their reproductivity? And then I'd also look at soil microbial community, obviously. I would monitor compaction. Where's my compaction depth? And depending on my resources, I would look at soil chemistry, and I would also look at that hand in hand with what are my plants taking up, what's inside of the plant. And I also look at bricks.
Is bricks part of that? What is that plant taking up?
That is part of photosynthesability, and there's all kinds of variation on how to do bricks responsibly, right? But so you pick your time of day and monitor your bricks as you change your land management practices and see what's happening. You can also, depending on what the yields are, what's my disease pressure, what's my insect pressure,